methionine is an I+ or minus amino acid with the chemical formula HO2CCH, NH2, CH2CH2SCH3. This amino acid is classified as nonpolar as it has a straight side chain that possesses S-methyl thioether at the I-cubed carbon. It is an essential amino acid in all metazoa. This amino acid is coded by the initiation codon AUG, which also indicates mRNA's coding region where translation into protein begins. Methionine, a proteinogenic amino acid, together with cysteine, methionine is one of two sulfur-containing proteinogenic amino acids. Excluding the few exceptions where methionine may act as a redox sensor, methionine residues do not have a catalytic role. This is in contrast to cysteine residues, where the thiol group has a catalytic role in many protein. The thioether does however have a minor structural role due to the stability effect of Si-Euro interactions between the side chain sulfur atom and aromatic amino acids in 1/3 of all known protein structures. This lack of a strong role is reflected in experiments where little effect is seen in protein with norleucine, and straight hydrocarbon side chain amino acid which lacks the thioether. It has been conjectured that norleucine was present in early versions of the genetic code, but methionine intruded into the final version of the genetic code due to the fact it is used in the cofactor S-adenosylmethionine. This situation is not unique and may have occurred with ornithine and arginine. In coding, methionine is one of only two amino acids encoded by a single codon in the standard genetic code. In reflection to the evolutionary origin of its codon, the other AUN codons encode isoleucine, which is also a hydrophobic amino acid. In the mitochondrial genome of several organisms, including metazoa and yeast, the codon AUA also encodes for methionine. In the standard genetic code, AUA codes for isoleucine and the respective tRNA uses the unusual base lysidine or agmatine to discriminate against AUG. The methionine codon AUG is also the most common start codon. A start codon is message for a ribosome that signals the initiation of protein translation from mRNA when the AUG codon is in a Kozak consensus sequence. As a consequence, methionine is often incorporated into the N-terminal position of proteins in eukaryotes and archaea during translation, although it can be removed by post-translational modification. In bacteria, the derivative N-formyl methionine is used as the initial amino acid. Methionine derivatives, S-adenosylmethionine. The methionine derivative S-adenosylmethionine is a cofactor that serves mainly as a methyl donor. SAM is composed of an adenosyl molecule attached to the sulfur of methionine, therefore making it a sulfonium cation. The sulfur acts as soft Lewis acid, allows the S-methyl group to be transferred to an oxygen, nitrogen or aromatic system, often with the aid of other cofactors such as cobalamin. Some enzymes use SAM to initiate a radical reaction, these are called radical SAM enzymes. As a result of the transfer of the methyl group, S-adenosyl homocysteine is obtained. In bacteria, this is either regenerated by methylation or is salvaged by removing the adenine and the homocysteine, leaving the compound dihydroxypentanedione to spontaneously convert into autoinducer 2, which is excreted as a waste product/quorum signal. Methionine as a biochemical precursor, this amino acid is also used by plants for synthesis of ethylene. The process is known as the Yang cycle or the methionine cycle. Biosynthesis, as an essential amino acid, methionine is not synthesized de novo in humans and other animals, who must ingest methionine or methionine-containing proteins. In plants and microorganisms, methionine biosynthesis belongs to the aspartate family, along with threonine and lysine. The main backbone is derived from aspartic acid, while the sulfur may come from cysteine, methanethyl or hydrogen sulfide. First, aspartic acid is converted via I2-aspartyl semialdehyde into homoserine by two reduction steps of the terminal carboxyl group. The intermediate aspartate semialdehyde is the branching point with the lysine biosynthetic pathway, where it is instead condensed with pyruvate. Homoserine is the branching point with the threonine pathway, where instead it is isomerized after activating the terminal hydroxyl with phosphate. Homoserine is then activated with an phosphate, succinyl or acetyl group on the hydroxyl. In plants and possibly in some bacteria, phosphate is used. This step is shared with threonine biosyntheses. In most organisms, an acetyl group is used to activate the homoserine. This can be catalyzed in bacteria by an enzyme encoded by mETX or meta. In enterobacteria and a limited amount of other organisms, succinate is used. The enzyme that catalyzes the reaction is meta, and the specificity for acetyl-CoA and succinyl-CoA is dictated by a single reside. The physiological basis for the preference of acetyl-CoA or succinyl-CoA is unknown, but such alternative routes are present in some other pathways. The hydroxyl activating group is then replaced with cysteine, methanethyl or hydrogen sulfide. A replacement reaction is technically AI-cubed elimination followed by a variant of Amical addition. All the enzymes involved are homologs and members of the cis-met metabolism PLP-dependent enzyme family, which is a subset of the PLP-dependent fold-type I-clade. They utilize the cofactor PLP, which functions by stabilizing carbanion intermediates. If it reacts with cysteine, it produces cysteoethernine, which is cleaved to yield homocysteine. The enzymes involved are cysteoethernine I-cubed synthase and cysteoethernine I-squared liase. Cysteoethernine is bound differently in the two enzymes, allowing I-squared or I-cubed reactions to occur. If it reacts with free hydrogen sulfide, it produces homocysteine. This is catalyzed by O-acetylhomoserine aminocarboxypropyltransferase liase. It is encoded by either mETY or mETs in bacteria. If it reacts with methanethyl, it produces methionine directly. Methanethyl is a byproduct of catabolic pathway of certain compounds, therefore this route is more uncommon. If homocysteine is produced, the thiol group is methylated, yielding methionine. Two methionine syntheses are known, one cobalamin-dependent and one independent. The pathway utilizing cysteine is called the transsulfuration pathway, while the pathway utilizing hydrogen sulfide is called direct sulfurylation pathway. Cysteine is similarly produced, namely it can be made from an activated serine and either from homocysteine or from hydrogen sulfide. The activated serine is generally O-acetylserine, but in aeropyrampernics and some other R-chiophosphazerine is used. Cis-K and cis-M are homologs, but belong to the PLP fold type 3 clade. Transsulfurylation pathway. Enzymes involved in the E. coli transsulfurylation route of methionine biosynthesis, aspartate kinase, aspartate semialdehyde dehydrogenase, homoserine dehydrogenase, homoserino-transsuccinylase, cysteoethernine I-cubed synthase, cysteoethernine I-squared liase, methionine synthase. Other biochemical pathways. Although mammals cannot synthesize methionine, they can still use it in a variety of biochemical pathways. Methionine catabolism. Methionine is converted to S-adenosylmethionine by methionine adenosyl transferase. SAM serves as a methyl donor in many methyltransferase reactions, and is converted to S-adenosylhomocysteine. 3. Adenosylhomocysteine converts SAH to homocysteine. There are two fates of homocysteine: it can be used to regenerate methionine, or to form cysteine. Regeneration of methionine. Methionine can be regenerated from homocysteine via methionine synthase in a reaction that requires vitamin B12 as a cofactor. Homocysteine can also be remethylated using glycine betaine to methionine via the enzyme betaine homocysteine methyltransferase. BHMT makes up to 1.5% of all the soluble protein of the liver, and recent evidence suggests that it may have a greater influence on methionine and homocysteine homeostasis than methionine synthase. Reverse transsulfurylation pathway. Conversion to cysteine. Homocysteine can be converted to cysteine. 5. Cysteoethernine I-squared synthase combines homocysteine and serine to produce cysteoethernine. Instead of degrading cysteoethernine via cysteoethernine I-squared liase, as in the biosynthetic pathway, cysteoethernine is broken down to cysteine and I+ or -ketabutyrate via cysteoethernine I-cubed liase. 7. The enzyme I+ or -ketoside dehydrogenase converts I+ or -ketabutyrate to propionyl-CoA, which is metabolized to succinyl-CoA in a three-step process. Chemical synthesis. Racemic methionine can be synthesized from diethyl sodium phthalimide myelinate by alkylation with chlorophyllmethyl sulfate E followed by hydrolysis and decarboxylation. Human nutrition. Dietary sources. High levels of methionine can be found in eggs, sesame seeds, Brazil nuts, fish, meats and some other plant seeds. Methionine is also found in cereal grains. Most fruits and vegetables contain very little of it. Most legumes are also low in methionine. However, it is the combination of methionine plus cysteine which is considered for completeness of a protein. Racemic methionine is sometimes added as an ingredient to pet foods. Methionine restriction. There is scientific evidence that restricting methionine consumption can increase lifespans in some animals. A 2005 study showed methionine restriction without energy restriction extends mouse lifespan. A study published in Nature showed adding just the essential amino acid methionine to the diet of fruit flies under dietary restriction, including restriction of essential amino acids, restored fertility without reducing the longer lifespans that are typical of dietary restriction, leading the researchers to determine that methionine OE acts in combination with one or more other EAAs to shorten lifespan. Several studies showed that methionine restriction also inhibits aging-related disease processes in mice and inhibits colon carcinogenesis in rats. In humans, methionine restriction through dietary modification could be achieved through a vegan diet. Veganism being a completely plant-based diet is typically very low in methionine, however certain nuts and legumes may provide higher levels. A 2009 study on rats showed methionine supplementation in the diet specifically increases mitochondrial ROS production and mitochondrial DNA oxidative damage in rat liver mitochondria, offering a plausible mechanism for its hepatotoxicity. However, since methionine is an essential amino acid, it should not be entirely removed from animals' diets without disease or death occurring over time. For example, rats fed a diet without methionine developed steatohepatitis, anemia and lost 2/3 of their body weight over 5 weeks. Administration of methionine ameliorated the pathological consequences of methionine deprivation. Methionine might also be essential to reversing damaging methylation of glucocorticoid receptors caused by repeated stress exposures, with implications for depression. Health. Loss of methionine has been linked to senile graying of hair. Its lack leads to a buildup of hydrogen peroxide in hair follicles, a reduction in tyrosinase effectiveness and a gradual loss of hair color. Methionine is an intermediate in the biosynthesis of cysteine, carnitine, taurine, lecithin, phosphatidylcholine, and other phospholipids. Improper conversion of methionine can lead to atherosclerosis. Other uses. D-L-methionine is sometimes given as a supplement to dogs. It helps keep dogs from damaging grass by reducing the pH of the urine. Methionine is allowed as a supplement to organic poultry feed under the U.S. Certified Organic Program. See also, allantoin, formylmethionine, methionine oxidation, paracetamol poisoning, a methionine paracetamol preparation that might prevent hepatotoxicity. Photoreactive methionine, references. External links, Rudra, M.N. Chowdhury, L.M. Methionine content of cereals and legumes. Nature 166, 568. Bibcode, 1950 Nature 166 568 doi, 10.1038/166568 AOA, food sources of methionine, foods containing methionine.